Hello there, and welcome. Take a slow, deep breath in, and gently breathe out. If this space helps you relax, consider subscribing and supporting the channel. Now, close your eyes, let your body relax, and let's begin. There's a kind of quiet that arrives just before dawn, when the night has not yet fully released its grip on the world, and the first gray light of morning touches the forest canopy from above, falling slowly through layer after layer of branch and leaf, until it finally reaches the ground. At that hour, in that particular stillness, if you were to lower yourself slowly from the height of the trees, past the eye level of the deer, below the reach of the ferns, all the way down until you are nearly flat against the earth, you would find yourself at the threshold of a world almost no one deliberately visits. It is a world measured not in miles, but in millimeters, a world where a raindrop becomes a lake, where the space between two small stones becomes a canyon, where a patch of ground no larger than your open hand contains dozens of individual plants growing so closely together that they form a single continuous surface. Soft and yielding, the color of patience itself. This is the world of moss. Dawn comes slowly here. The light does not arrive as a flash or a flood. It seeps in, the way water seeps into dry soil, gradually, unhurried, and as it does, the moss begins to glow, not in any dramatic way, not like a lamp being switched on, or a fire catching hold. It glows the way something glows when it has been waiting, when it has been quietly absorbing and holding and existing in the dark, and the light simply allows you to see what was always there. The color is remarkable, not simply green. There are 100 greens in a single square foot of forest moss. The deep, almost black green of shadow-soaked clumps beneath overhanging rocks. The bright, almost yellow green of fresh growth along a stream bank. The muted, dusty green of a colony in its dormant phase, waiting out a dry spell with no particular urgency. Greens that exist nowhere else in nature, so specific to this combination of light and water and time that they resist description. For most of human history, we have walked past this world without noticing it. We are a species oriented toward height. We look up at trees, at mountains, at the sky. We measure progress in elevation. We build upward. We aspire upward. The word upward carries with it an entire architecture of meaning, advancement, aspiration, hope. And so the world beneath our feet goes unexamined, walked across, compressed beneath boot soles and tire treads, and the casual inattention of a species too busy looking at the horizon to notice what lies beneath. But here is a question worth sitting with, quietly, as the morning light continues to arrive. What if the quietest plants on earth shaped the land before forests ever rose? What if, long before the first tree stretched its roots into soil, before the first fern uncoiled its fronds in the carboniferous air, before anything we would recognize as a landscape existed on land at all? What if something very much like moss was already there, working at the surface of the bare rock, inch by inch, century by century, doing the slow, patient work of turning stone into ground? That is not a poetic exaggeration. It is, in careful and considered terms, what the fossil record and the study of living plants suggests. The ancestors of today's mosses, small, rootless, water-dependent plants, are thought to be among the first photosynthesizing organisms to establish themselves on land. They did not conquer the earth with dramatic flourish. They simply arrived, held on, and began the long, quiet work of changing everything around them. And they are still doing it. This documentary is an invitation, not to dramatic revelation, not to sudden astonishment, but to something more deliberate. The act of lowering yourself to ankle height and looking carefully at what has always been there. The act of noticing. The act of taking seriously the smallest, most quietly persistent forms of life. Kneel down. Let the damp coolness of the forest floor come through the fabric of your clothes. Hold your hand flat and press it gently against a patch of moss. Feel the give of it, that soft resistance, that cushion of green that yields beneath your fingers as though it were something alive and responsive. Which it is, which it absolutely is. Take a breath. Notice the smell, that particular combination of earth and water and green that exists nowhere else. That complex, quiet fragrance of living moss in damp shade. A scent so specific that it arrives in the brain not merely as information, but as something older, something that triggers a response too deep and wordless to fully name. Now stay here for a while, because what follows is not a story about small things that don't matter. It is a story about small things that shape the world. And there is a difference. To understand moss, it helps to begin by setting aside everything you think you know about plants. When most of us picture a plant, we picture something with roots that reach down into the soil, that anchor the plant in place, that pull water and dissolved minerals upward through a network of internal tubes. A system not entirely unlike the plumbing in a building, where pipes carry essential resources from one place to another. We picture stems that hold the plant upright, leaves that capture sunlight, flowers perhaps, or seeds. We picture a plant as a structured, hierarchical organism, with distinct parts performing distinct functions. Moss does not work this way. Moss belongs to a group of plants called bryophytes, a word derived from the Greek for moss plant, though the group also includes liverworts and hornworts. Bryophytes are, in the broadest sense, the simplest of all land plants. Not simple in the sense of primitive or inferior. Simplicity in biology often represents remarkable efficiency, but simple in structure, in the way that a well-designed tool is simple. It does exactly what is needed, with nothing wasted. Mosses have no true roots. This is perhaps the most fundamental thing to understand about them. And it requires some unpacking, because they do have structures that look like roots, that occupy the same space in the ground where roots would be, and that perform some of the same functions. These structures are called rhizoids, and they are worth understanding carefully, because they reveal something essential about how moss relates to the world around it. Rhizoids are thin, thread-like filaments that anchor the moss to whatever surface it is growing on. Rock, bark, soil, old wood, the surface of a wall. They hold the plant in place. They prevent it from being washed away by rain or blown away by wind. In some mosses, research suggests they may also absorb small amounts of water and nutrients directly, but they are not true roots in the botanical sense. True roots are vascular structures. They contain specialized tissue that transports water and nutrients throughout the plant. Rhizoids lack this internal transport system. They are, in a sense, anchors without pipes. And this leads to the second fundamental truth about mosses. They have no vascular tissue. Vascular tissue is the internal plumbing of most land plants. The xylem that carries water upward from roots to leaves, and the phloem that carries the products of photosynthesis downward from leaves to roots and throughout the plant. This system allows tall trees to pull water from the soil to their highest branches, allows sprawling vines to transport resources across dozens of meters, allows the enormous diversity of flowering plants to build complex bodies with specialized organs. Mosses have none of this. There are no internal tubes, no specialized transport cells, no long-distance movement of water and nutrients through the body of the plant. Some mosses do have simple conducting cells, structures that look a little like vascular tissue and perform some similar functions. But these are far simpler than true vascular tissue, and are absent in many species entirely. This means that each part of the moss must largely take care of itself. Water does not travel from a root system up through a stem to the leaves. Instead, each leaf, if we can call it that, since moss leaves are not true leaves in the botanical sense, absorbs water directly from its surface. Rain falls on the moss. Dew condenses on its surfaces in the cool of the morning, and the cells of the plant absorb that water directly through their outer walls. The plant is, in essence, drinking through its skin. This is why moss is so dependent on moisture. It cannot reach down into the soil for water the way a tree can. It cannot pull up a reserve of groundwater during a dry spell. When moisture is available, when it rains, when dew forms, when the air itself is saturated with humidity, moss absorbs it eagerly and fills its cells. When moisture is absent, the moss dries, its cells contract, the plant goes dormant, waiting. We will return to this remarkable capacity for patience and recovery later. For now, consider what it means to live without roots, without an internal plumbing system, without the structural complexity that most land plants take for granted. Consider what it demands of a plant, and what it requires it to give up. What it demands is proximity. Moss must live close to its water source, close to the air's moisture, close to the surface of streams, in the humid shade beneath tree canopies, in the permanently damp crevices of rocks. It cannot afford to be far from water, because it has no way of reaching for water it cannot immediately touch. What it requires giving up is height. Without vascular tissue, a plant cannot pump water upward against gravity to feed a tall structure. Moss stays low, because its body structure requires it to stay low. The tallest mosses in the world are only a few centimeters high. Most are much shorter. This is not failure. It is the consequence of a body plan that chose, if we can use such a word of evolution, efficiency and resilience over ambition and height. The individual leaves of a moss, examined under magnification, reveal a different kind of architecture than the complex leaf structure of a flowering plant. Many moss leaves are just a single cell thick. You read that correctly. One cell thick. Light falls on one surface and exits from the other. Every cell participates equally in photosynthesis. There is no specialization into layers of different cell types. No waxy cuticle, no stomata, no spongy mesophyll, no palisade layer, just a single transparent sheet of living cells, each one open to the environment, absorbing water, conducting photosynthesis, living in direct contact with the world around them. This design is old, very old. It reflects an architecture that has been working, without substantial modification, for hundreds of millions of years. When something works this well, for this long, in this many different environments, from tropical rainforests to Arctic tundra, it is worth taking seriously. Mosses are not failed trees. They are not protoplants waiting to evolve into something more complex. They are exactly what they are. A lineage that found a way to live on land without the complexity that most other plants require. And that has persisted in that way, in enormous variety, through every dramatic change the Earth has experienced since the Devonian period. There are roughly 12,000 species of moss currently recognized by science. They live on every continent, including Antarctica. They grow on the surfaces of other plants, on bare rock, on the bark of trees, in the beds of streams, in the spray zones of waterfalls, in the cracks of city pavements, on old gravestones, on the north sides of fence posts, on the tile roofs of ancient buildings. They are, in the fullest sense of the word, everywhere. And yet, we walk past them every day, without noticing. Imagine the Earth, as it was approximately 450 million years ago. Not the lush green planet we know today. That Earth did not yet exist. The seas were full of life. Early fish moving through warm, shallow water. Algae covering the ocean floor in great mats. Complex communities of invertebrates building reef structures and crawling across the sediment. The oceans were alive, but the land was largely bare. Not entirely bare. There is evidence that early microbial life colonized land surfaces long before plants arrived, forming thin biological crusts on rock and soil. But the land as we would recognize it today, covered in plants rooted in deep soil, shaded by canopies of trees, did not yet exist. There were no forests. There were no prairies. The continental interiors were likely vast, dry, and largely empty, scraped by wind and scoured by occasional rain. Their rock surfaces the color of the rock itself, with no green anywhere. And then gradually, that began to change. The precise details of how plants first colonized land remain an active area of scientific research. And the story is considerably more complex than any brief telling can fully convey. What scientists broadly agree on is that the first land plants evolved from freshwater green algae, specifically from a group called the charophytes, which still exist today in ponds and streams. These algae were already adapted to shallow water environments, where they sometimes experience periods of partial exposure to air. Over an enormous span of time, millions upon millions of years, some of these organisms developed adaptations that allowed them to survive increasingly prolonged exposure to dry conditions, to direct sunlight, and to the physical challenges of a world without the buoyancy and constant moisture supply of water. The earliest land plants, as best as the fossil record can tell us, were small, low-growing, and rootless, resembling, in broad terms, modern liverworts and mosses. These plants did not need to be tall. The land was open, unshaded, uncrowded. There was no competition for height, because nothing else was tall. What was needed was the ability to cling to rock surfaces, to withstand desiccation during dry periods, to capture water from rain and dew, to grow slowly across exposed surfaces. And this is what moss-like plants did. The fossil evidence for early land plants is fragmentary and often difficult to interpret. Plant tissues do not preserve easily under most conditions. But what evidence we have, combined with the study of living plant lineages and their genetic relationships, suggests that the bryophytes, the group that includes mosses, liverworts, and hornworts, represent some of the earliest branches of the land plant family tree. Research suggests they diverged from the ancestors of all other land plants, very early in the history of terrestrial life. What does this mean in practical terms? It means that long before the first tree evolved, long before the first fern, or the first seed plant, or the first flowering plant, there were plants very much like today's mosses covering the surfaces of rocks and soil, doing the slow work of photosynthesis, building up organic matter, beginning the process of changing bare mineral surfaces into something that could eventually support more complex life. Consider what that process actually involves. When moss grows on bare rock, it does not simply sit there passively. Its rhizoids penetrate tiny cracks and irregularities in the rock surface. And as the plant grows and dies and grows again, organic material accumulates. The acids produced by the moss, simple organic acids, not dramatically corrosive, but persistent, very slowly break down the mineral surface of the rock. Water absorbed by the moss expands when it freezes, widening cracks. Dead moss tissue accumulates, mixing with mineral particles, beginning to form the first thin layer of what might eventually become soil. This is a process measured not in years or decades, seconds, but in centuries, in millennia, it is so slow as to be invisible on any human timescale. But it is happening, right now, on every moss-covered rock surface in every forest and mountain and tundra on Earth. This process is proceeding at its own unhurried pace. Soil does not simply exist. It is made. It is the product of biology acting on geology. Over time, organisms processing rock and organic matter into the complex living medium that most land life depends on. And moss, along with its ancestors, was among the first organisms to participate in that making. The forests that cover so much of the Earth today, the agricultural soils that feed billions of people, the grasslands and wetlands and meadows, all of them rest ultimately on foundations that began to be laid hundreds of millions of years ago by plants like moss. It is worth sitting with that for a moment. By the time trees evolved, the first truly tall, woody vascular plants appeared perhaps 360 million years ago in the late Devonian period. The world had already been profoundly changed by the small, rootless, water-dependent plants that had come before them. There was soil where there had been bare rock. There was organic matter in that soil, and with it the beginnings of the complex soil communities, bacteria, fungi, early invertebrates, that vascular plants would need to thrive. Trees did not colonize a bare world. They colonized a world that had been prepared for them. When the first trees rose up, their woody trunks lifting photosynthesizing leaves above the competition, their roots reaching down through soil layers that had been accumulating for millions of years. Moss was already there, not competing with the trees, not struggling against their shade, but simply continuing to do what it had always done, growing close to the ground, absorbing water from rain and dew and morning fog, building up organic matter, maintaining the surface of the world. And it is still there. Walk into any temperate forest today and look down, beneath the trees, at the base of their massive trunks, across the surfaces of the rocks and fallen logs, covering the soil between the roots. The moss is there, older, in evolutionary terms, than the trees themselves, outlasting, in some lineages, entire geological epochs that have come and gone, patient beyond any human understanding of patience. This is not a fairy tale about an ancient plant with mystical powers. It is simply the factual history of life on land, which contains within it a quiet and rather astonishing truth. The smallest, simplest things were first, and they are still here. To understand how moss drinks, it helps to think about physics before thinking about biology. Specifically, it helps to think about the behavior of water when it encounters very small spaces. You have seen this behavior, even if you have never named it. It is what happens when you touch the tip of a piece of paper to the surface of a puddle, and the water climbs up into the paper without being pulled by any visible force. It is what happens in the thin tube of a thermometer, where mercury or colored alcohol moves upward against gravity. It is what makes a candle work, the liquid wax moving up through the wick toward the flame. This phenomenon is called capillary action, and it is one of the most fundamental tools in the moss toolkit. Capillary action arises from two properties of water working together. The first is adhesion. Water molecules are attracted to the surfaces of certain materials, paper, fabric, plant tissue, and they cling to those surfaces. The second is cohesion. Water molecules are attracted to each other, forming a kind of chain. When water adheres to a surface at the bottom of a narrow space, it pulls more water behind it, and the cohesion of water molecules means that pull travels upward. In very narrow spaces, the adhesion can overcome gravity, and water climbs. Moss has evolved a body plan that takes extraordinary advantage of this physics. The structure of a moss colony, those densely packed stems and leaves, the minute spaces between individual plants, the overlapping arrangements of tiny leaves along each stem, creates an enormous network of capillary spaces. Water that falls on the surface of a moss colony, or that arrives as dew, or that seeps up from a saturated substrate, enters this network and is drawn through it by capillary action, moving between and among the stems and leaves, without any need for an internal pumping system. Different species of moss have developed different structural approaches to water capture and retention, and the variety is remarkable. Some mosses have leaves arranged in patterns that funnel water toward the stem, channeling each raindrop efficiently toward the place where absorption is most effective. Others form such densely packed mats that the air spaces within the colony are extremely small, and capillary forces correspondingly strong, allowing these mosses to hold large amounts of water even between rainfall events. Some species have evolved leaves within large, dead, water-holding cells interspersed among the living photosynthetic cells. In the genus Sphagnum, the peat mosses, this strategy is taken to an extreme. Up to 2/3 of the volume of a sphagnum leaf may consist of these large, empty, structural cells, called hyaline cells, that fill with water and hold it like a sponge. A living sphagnum mat can hold many times its own dry weight in water. We will return to sphagnum at length when we reach the section on peat bogs. But even beyond sphagnum, the relationship between moss and water is one of profound intimacy and mutual shaping. Water is not just a resource for moss. It is, in a real sense, the medium in which moss lives, the material around which its entire body plan is organized, the substance that connects it to every other part of its environment. Think about what happens in the minutes after a rain shower arrives in a forest. The rain falls first on the canopy, high above, a sound like many small hands patting leaves, irregular and shifting. Most of the rain is intercepted up there by the broad leaves of the canopy trees and understory shrubs. Some of it drips from leaf tip to leaf tip, gradually working downward. Some of it runs down the bark of tree trunks in thin sheets. Only a portion of it reaches the forest floor directly. But eventually, it does arrive at the floor. It arrives as large drops falling from overhanging leaves, drops that have coalesced from many smaller drops during their passage through the canopy. It arrives as runoff from bark and branches. It arrives during prolonged rain, as a general saturation of the forest atmosphere, a fog of moisture suspended in the air beneath the canopy. And when it arrives at the moss, something remarkable happens that is visible if you are watching closely. A dry or partially dry moss colony, one that has been waiting through a dry period. Its leaves contracted, its cells shrunken, undergoes a visible transformation when water arrives. The leaves, which in the dry state may have been curled tightly against the stem, or folded in various protective configurations, begin to open. They unfurl slowly, like something waking up. As the cells absorb water and expand, the color deepens. The surface of the colony becomes more reflective, catching what light filters through the canopy. And then the absorption begins in earnest. Water moves through the capillary network of the colony, spreading from where it lands outward and downward, being absorbed by cells across the colony's surface. The colony may absorb a substantial portion of its own weight in water within minutes of rainfall beginning. This is not a metaphor for efficiency. It is simply what the structure of moss makes possible. On steep slopes, this rapid absorption serves an important function for the broader landscape. Rain that falls on bare soil runs off quickly, carrying soil particles with it. Rain that falls on moss is absorbed and held. The flow of water off the slope is slowed, The water is retained in the plant tissue and released gradually. As it evaporates or seeps downward into the soil, the moss acts as a buffer between the rain and the mineral substrate beneath it, moderating the intensity of what the soil has to absorb. There is also the matter of dew. In many environments, dew represents a significant water source, particularly in regions where rainfall is infrequent. But nighttime temperatures regularly drop enough to cause condensation on cool surfaces. Moss is well positioned to capture dew. Its large surface area relative to its volume, all those tiny leaves, all those exposed surfaces, provides ample area for condensation to form. By morning, a moss colony that received no rain may nonetheless glitter with dozens or hundreds of tiny water droplets, each one resting in the cup of a leaf, or hanging from a leaf tip, each one slowly being absorbed as the moss warms through the morning hours. Watch this happen. If you have the opportunity to be in a forest or garden at dawn, before the sun has had a chance to warm the surface, and before any wind has disturbed the dew, lower yourself to the level of the moss and look across it at a shallow angle. The droplets are everywhere, each one a perfect sphere, each one containing a tiny inverted image of whatever is above it. They rest there for only a short time, before being absorbed or evaporated, but in that brief window, the moss colony has captured and begun to consume a water source that would have been entirely unavailable to most other plants in the same environment. This relationship with water, the dependence on it, the exquisite structural adaptation to capture and hold it, the vulnerability that comes from having no internal reserves except what the plant body itself can contain, defines what moss is and what it can and cannot do. It is simultaneously a limitation and an extraordinary capability. A limitation because it tethers moss to moist environments and leaves it vulnerable to extended dry periods. A capability because it allows moss to exploit water sources and environmental conditions that other plants cannot effectively use. The sound of rain on moss is different from the sound of rain on bare soil. Softer, more absorbent, as though the moss were consuming the sound along with the water, pulling both into itself, making both part of what it is. The life cycle of moss is a story about water at every stage. To understand it fully requires setting aside the familiar template of most plant reproduction. The flowers, the pollen, the seeds, the fruit, and encountering something older, stranger, and in its own way more interesting. Most of the moss you see when you look at a moss colony, the green, leafy, photosynthetic plant that covers rocks and logs and forest floors, is technically only half the plant. Or rather, it is one phase of a two-phase life cycle, so fundamental to the biology of mosses and other early land plants, that it has its own name, alternation of generations. This concept is easiest to understand by starting with what is familiar. In flowering plants, the plant body you see, the tree, the flower, the grass, is the sporophyte generation, the phase of the life cycle that produces spores. But in a flowering plant, the sporophyte phase so completely dominates the life cycle that the other phase, the gametophyte, is reduced to just a few cells hidden inside the flower. It has been miniaturized almost to nothing, a vestigial echo of an older way of living. In mosses, the balance is reversed. The dominant phase of the life cycle, the green leafy plant that forms the carpet and colony and cushion that we recognize as moss, is the gametophyte. The word comes from the Greek for marriage plant, because the gametophyte is the phase that produces the gametes, the sperm and the eggs, that will combine to create a new plant. The gametophyte phase of a moss is haploid, meaning its cells each contain only one set of chromosomes, half the genetic complement of a human cell or a flowering plant cell. This is the normal state for gametes in most organisms. But in mosses, it is the state of the entire plant body. Every cell in the leafy moss you are looking at contains only a single set of chromosomes. When conditions are right, and here, crucially, water enters the story again. The gametophyte produces reproductive organs. Male gametophytes produce structures called antheridia that release sperm cells, and female gametophytes produce structures called archegonia that contain the egg cells. In some mosses, both types of reproductive structures occur on the same plant. In others, they occur on separate plants of different sexes. The sperm cells of mosses are motile. They swim. This is a fact that connects moss more directly to its aquatic ancestors than almost any other aspect of its biology. The sperm of a moss is structurally similar to the sperm of many aquatic algae, bearing one or more flagella that they use to propel themselves through water. And this is why moss reproduction requires water. Without a film of water through which the sperm can swim from the antheridium to the archegonium, fertilization cannot occur. This water film, sometimes as thin as the surface tension on a wet stone, sometimes as substantial as a temporary puddle left by rain, sometimes simply the damp surface of a moss colony after a heavy dew, is the medium of reproduction. On a rainy day, or in the hours after rain has fallen, moss sperms swim through this film in their millions, navigating by chemical gradients toward the archegonia that contain the eggs. The distances they travel are tiny in absolute terms, a few millimeters, perhaps a few centimeters. But relative to the size of the sperm, these are extraordinary journeys, and the sperm that successfully reaches an egg and fertilizes it creates something new, a diploid cell containing two sets of chromosomes, which will grow into the second phase of the moss life cycle. The sporophyte phase of moss looks nothing like the gametophyte. It does not form the green leafy carpet that we associate with moss. Instead, it grows upward from the fertilized egg as a thin stalk, topped by a structure called a capsule, which is where spore production occurs. The sporophyte is dependent on the gametophyte for much of its nutrition. It cannot survive independently, but grows attached to and sustained by the leafy plant that produced it. Walk slowly through a forest in late spring or early summer, and look closely at the moss growing on rocks and logs. In many species, you will see what look like tiny upright stalks rising from the surface of the moss colony, each one topped by a small oval or cylindrical capsule. These are the sporophytes. Depending on the species, these capsules may be pale green, or reddish brown, or almost black. They may stand upright, or droop gracefully to one side. They are in their own way beautiful. These minimal structures, rising above the horizontal world of the gametophyte colony like small exclamation marks. Inside each capsule, meiosis produces spores, cells that, like the gametophyte, are haploid, and that will grow into new gametophyte plants if they land on a suitable surface. The number of spores produced by a single capsule varies by species, but it is often large, many thousands, sometimes more. This abundance compensates for the extraordinary inefficiency of dispersal. Most spores will land somewhere unsuitable and fail to develop. Only a tiny fraction will find a surface with the right combination of moisture, light, and substrate to allow germination. The release of spores from a mature capsule is a subtle, unhurried process. The capsule, as it matures, develops a structure at its opening, a ring or fringe of small teeth-like projections called the peristome, that regulates spore release in response to environmental conditions. In dry conditions, the teeth close over the capsule opening, retaining the spores. In humid conditions, They open, allowing spores to escape into the air. This mechanism is thought to favor spore release during conditions that are favorable for dispersal. Humid air tends to allow spores to remain airborne for longer periods, and the air currents associated with changing humidity may carry spores further from the parent plant. When a spore lands on a suitable surface, and conditions are right, it begins to germinate. The first structure to form is not a small version of the adult moss plant, but a thread-like green filament called the protonema, another structure that resembles the filamentous algae of the aquatic world that moss's ancestors came from. The protonema spreads across the surface of the substrate, photosynthesizing, establishing the plant's presence in its new location. Eventually, from this filamentous mat, the familiar leafy shoots of the gametophyte emerge and begin to grow. The entire cycle, from spore to mature sporophyte producing new spores, takes varying amounts of time depending on species and conditions. Typically anywhere from 1 to several years for the full cycle to complete. This is not slow exactly, but neither is it fast. It is the pace of moss, deliberate, methodical, unhurried by any urgency we can understand. There is something worth pausing over in this life cycle. The central role of water at every stage. Water for the capillary transport of nutrients. Water as the medium through which sperm cells swim to find eggs. Water absorbed by spores to allow germination. Water absorbed by the protonema and gametophyte throughout their development. The entire existence of the moss, from birth to reproduction to death, is wound around water as a vine winds around a fence post. Completely, thoroughly, at every point of contact. This is the inheritance of an aquatic ancestry, carried forward into the terrestrial world, but never entirely shed. Moss lives on land, but it carries the ocean with it, in the architecture of its body, and in the requirements of its reproduction. And perhaps that is part of what makes the sound of rain falling on moss feel, to those who listen carefully, like something more than just weather. It is, in a quiet way, the sound of a life cycle completing itself. If you were to borrow the eyes of something very small, a mite perhaps, or a springtail, or a rotifer, and use those eyes to look at a patch of moss from within, you would not see a carpet or a cushion. You would see a forest. The stems of the moss plants, rising centimeters above the substrate, would appear as tree trunks, their surfaces textured and branching. The leaves would form a canopy above, a dense, overlapping ceiling of green, through which light filters in patterns that shift as the canopy moves in a breeze. Between the stems, there would be open spaces, tunnels, chambers. The microarchitectural complexity of a habitat that has been organizing itself over years or decades of accumulated growth. And in every part of this microforest, life would be everywhere. The invertebrate community living within a healthy moss colony can be surprisingly diverse. Mites, which belong to the same group as spiders, are typically present in large numbers, feeding on algae, fungal spores, dead plant material, and each other. Springtails, small wingless hexapods with a forked structure that allows them to leap, move through the spaces between moss stems, also feeding on decaying organic matter and fungal material. Nematodes, microscopic roundworms among the most numerically abundant animals on Earth, thread through the water films that coat moss tissue and the substrate beneath, feeding on bacteria and other microorganisms. And then there are the tardigrades. Tardigrades, also called water bears or moss piglets, are microscopic animals, typically less than 0.5 millimeter long. They have 8 stubby legs, a barrel-shaped body, and a feeding apparatus designed to pierce plant cells and consume their contents. Under a low-powered microscope, they appear almost endearing, slow, rounded, methodical creatures, moving through the water film on moss leaves with unhurried deliberateness. Tardigrades have attracted scientific attention well beyond their size because of their extraordinary resistance to environmental extremes. When conditions become unfavorable, when the moss dries out, when temperatures drop toward freezing, when oxygen levels fall, tardigrades can enter a remarkable state called cryptobiosis. In this state, their metabolic activity drops to nearly zero. Their body loses almost all its water, and they persist in a kind of suspended animation. When moisture returns, they rehydrate and resume their lives. But let us not get lost in the remarkable and miss the ordinary ecology of a moss colony, the day-to-day, unremarkable community of organisms going about their business, is itself worth paying attention to. A moss colony retains moisture. This is fundamental to its body plan, but the ecological consequence is profound. Temperatures within the colony fluctuate less than at exposed soil surfaces. The moss insulates, moderating extremes of heat and cold. Humidity remains higher for longer after rainfall. The interior surfaces are cool, dark, and damp long after the surrounding landscape has dried. These conditions are exactly what many small invertebrates need. Moisture-dependent animals, those without a waterproof cuticle, or those that reproduce through aquatic intermediary stages, find in moss habitats a refuge that may be unavailable in surrounding drier environments. The moss colony becomes, in effect, an island of suitable conditions in a landscape of variability. Consider the community at different scales. At the microscopic level, bacteria, archaea, and fungi decompose accumulating organic matter. Algae and cyanobacteria grow on the surfaces of moss leaves, contributing to photosynthesis. Protozoa prey on bacteria. Nematodes and rotifers move through water films. At the slightly larger scale, tardigrades, mites, and springtails occupy the spaces between stems. Larger still, beetle larvae, centipedes, and isopods move through the deepest, most decomposed lower layers of older colonies. Some of these organisms contribute to the decomposition of dead moss tissue. The lower layers, parts that were living years or decades ago, die and begin to break down. Decomposition releases nutrients back into the substrate, making them available to the living moss above and to the roots of nearby vascular plants. The moss colony is not simply a passive backdrop. Its architecture determines the structure of the habitat, the size of the spaces between stems, the depth of the water films on leaves, the depth to which light penetrates. As the moss grows and changes, the habitat changes with it. Newly grown tips, established middle sections, decomposing bases. Each offers different conditions. Each supports different communities. The colony is not uniform, but layered, a gradient of conditions across depth and width, creating niche space for a surprising variety of organisms. There are also intimate relationships between moss and fungi. Many mosses appear to host fungi within their tissues, relationships that may be mutualistic, the fungus providing some benefit to the moss in exchange for access to photosynthetic products. The nature of these associations is not fully understood. Research continues, but their presence suggests that the moss colony is not merely a gathering of distinct organisms living side by side. It may be, at times, a more integrated assemblage, with boundaries that blur. This is, in a sense, the moss colony as a metaphor for ecology itself. Apparent simplicity on the surface, concealing intricate complexity beneath. A square foot of forest moss examined carefully contains more species than most people encounter in a week of ordinary life. Not dramatic species, not species with common names, Not species with starring roles in nature documentaries, but species nonetheless, living, feeding, reproducing, dying, transforming matter from one form to another, participating in the continuous exchange of energy and materials that constitutes the living world. Small, quiet, everywhere. Soil seems like it should be simple. It is, after all, just dirt, the ground we walk on, the material gardeners dig in, the substrate from which plants grow. But soil is one of the most complex materials on Earth, and its formation is one of the slowest and most consequential processes in the natural world. True soil, the kind that supports diverse plant communities, that holds water and nutrients, that teems with the microbial life that makes nutrients available to plants, does not simply exist. It develops over long periods through the interaction of physical processes, chemical weathering, biological activity, and the accumulation of organic material. And early in that process, before vascular plants with their complex root systems can play their part, simpler organisms do the foundational work. Moss, along with lichens, cyanobacteria, and other members of the biological soil crust community, participates in the very early stages of soil development on bare rock and mineral substrates. This is not a process with a visible dramatic impact. You cannot watch it happening in any meaningful way. But over the spans of time that geology operates in, it is a process with profound consequences. The physical contribution of moss to soil formation involves several mechanisms. As we have discussed, rhizoids penetrate the surface of rock and other substrates, and as the plant grows and the rhizoids expand, they can create small fractures and increase the surface area available for chemical weathering. Water absorbed by moss expands when it freezes in winter, exerting physical force on cracked surfaces and gradually widening them. The alternating wetting and drying cycles associated with a moss colony, the surface beneath the moss being repeatedly wetted and then partially dried, contribute to the physical weathering of the substrate. The chemical contribution involves the organic acids produced by living and decomposing moss tissue. These acids, simple organic molecules released as byproducts of metabolism, react with minerals in the substrate, slowly breaking them down, and releasing mineral ions that may be taken up by the moss or become part of the developing soil. The process is gentle and slow, but it is continuous, and over centuries it produces a measurable effect on the mineral surface beneath a moss colony. But perhaps the most significant contribution of moss to soil formation is simply the accumulation of organic matter. Moss grows, and when it dies, or more precisely, when the lower portions of a moss colony die as the colony grows upward, the dead tissue accumulates on the substrate surface. This organic material is the foundation of soil. Mixed with mineral particles from the weathered substrate beneath, it provides the organic component of developing soil. The carbon compounds, the nitrogen, the complex of decomposing materials that soil microorganisms need to build a living community. As the soil community develops, the conditions for more complex plant life improve. Small vascular plants, mosses of a different kind, or early ferns or tiny annuals, may begin to establish themselves in the accumulating soil. Their roots penetrate and further loosen the substrate. When they die, they add more organic material. The soil deepens millimeter by millimeter, century by century. It is important to be accurate about the timescales involved here. The formation of deep, mature soil from bare rock is not a process that occurs in decades or even in centuries, but typically in thousands to tens of thousands of years. The moss is not dramatically transforming the landscape in any human perceivable time frame. What it is doing is contributing, persistently, and without interruption, to a process whose cumulative effects are significant on geological timescales. In addition to this long-term soil-building role, moss also plays a more immediately observable role in soil stabilization. Moss colonies growing on soil surfaces hold that soil in place. The rhizoids penetrate the upper layers of the soil, binding them together. The above-ground mat of stems and leaves intercepts rainfall and reduces its erosive impact on the soil surface beneath. In environments where soil erosion is a potential problem, steep slopes, areas with limited vegetation cover, recently disturbed sites, moss cover can make a meaningful difference in how much soil is retained. This stabilization function is particularly evident in the early stages of ecological succession after disturbance. After a forest fire, for example, the bare soil surface is exposed to rainfall and wind without the protective covering of vegetation. Mosses, which can colonize bare soil relatively quickly, they do not need to establish an extensive root system, and their spores are widely dispersed, are often among the first plants to reestablish cover on burned surfaces. As they do, they begin to moderate the temperature and moisture conditions at the soil surface, reduce erosion, and begin to rebuild the soil organic matter that was consumed by the fire. This is not to make extravagant claims about the centrality of moss to ecosystem recovery. Many other organisms participate, and the specific patterns of succession after disturbance vary enormously depending on climate, soil type, and the nature of the disturbance. But moss's contribution to soil stabilization and early successional processes is real and observable, not merely theoretical. There is also the relationship between moss and the mycorrhizal fungi that associate with the roots of vascular plants. These fungi form networks through the soil, extending the effective reach of plant roots, and facilitating the uptake of phosphorus and other nutrients. Research suggests that the decomposition of moss in forest soils contributes to the pool of organic matter and nutrients that the mycorrhizal network draws on. In this way, the moss's contribution to the soil community may extend beyond its own direct effects to influence the larger network of plant-fungal relationships that characterizes forest ecosystems. All of this, the slow weathering of rock, the accumulation of organic matter, the stabilization of soil surfaces, the creation of conditions suitable for the soil microbial community, is work done without intention, without awareness, without effort in any sense that we could meaningfully apply to consciousness. The moss does not build soil because it means to. It builds soil because it grows, because it is made of organic material that decomposes when it dies, because its physical structure interacts with its environment in ways that have cumulative effects. The soil beneath a forest that has been covered in moss for 1,000 years is different from the soil beneath a forest that has been covered in bare mineral substrate for the same period. Deeper, richer in organic matter, more capable of holding water, more diverse in its microbial community, more capable of supporting the complex web of plant and animal life above it. And beneath the moss, mostly unseen and unnoticed, that difference continues to accumulate. There is a genus of moss so different from most other mosses, so ecologically significant, so structurally remarkable, that it deserves its own careful consideration. The genus is Sphagnum, commonly known as peat moss, and the ecosystems it creates and inhabits, the bogs, fens, and mires of the world's temperate and boreal regions, are among the most distinctive and scientifically interesting habitats on Earth. To begin with the biology, sphagnum is distinguished from most other mosses by the structure of its leaves. Each sphagnum leaf contains two types of cells arranged in a specific pattern. living green photosynthetically active cells called chlorophyllose cells, and much larger, dead transparent cells called hyaline cells. The hyaline cells have gaps in their walls through which water can enter and leave. And the interior of the cell is essentially hollow, a tiny vessel designed for water storage. The ratio of hyaline to chlorophyllose cells in sphagnum leaves means that the leaf is largely made up of empty space that can be filled with water. This gives sphagnum an extraordinary water-holding capacity. A living sphagnum mat can hold very large amounts of water relative to its dry weight. The exact figures vary by species and conditions, but the capacity is substantial, which is why the plant is used in horticulture as a soil amendment and moisture retainer. But the ecological significance of sphagnum goes well beyond its structural properties. Sphagnum actively modifies its environment in ways that make it hostile to most other plant species, but highly favorable to itself, a phenomenon sometimes called ecosystem engineering. Sphagnum releases hydrogen ions as it grows, acidifying the water around it. Bogs dominated by sphagnum typically have water pH values well below neutral, in the range of 3.5 to 4.5, acidic enough that most nutrient-cycling bacteria cannot function effectively. The result is that the organic matter, dead sphagnum tissue, that accumulates in a sphagnum bog does not decompose the way organic matter normally would. Decomposition is severely slowed. When organic matter doesn't decompose, it accumulates. Over thousands of years in sphagnum-dominated bogs, dead plant material has been accumulating layer upon layer, compressing under the weight of new growth above, gradually becoming the dense, dark, fibrous material we call peat. Peat is essentially partially decomposed organic matter, a physical record of centuries of plant growth that was never fully broken down. Peat deposits can be very deep. In some northern bogs and fens, peat accumulates to depths of many meters, representing thousands of years of accumulated organic material. Carbon, which all plants contain as the fundamental building block of organic chemistry, is locked up in this peat in very large quantities. Because the carbon in the dead sphagnum is not being decomposed and released back into the atmosphere as carbon dioxide, it remains stored in the peat. Research suggests that the world's peatlands store a significant proportion of the carbon found in soils globally. Estimates vary, but the numbers cited in scientific literature are consistently substantial. This carbon storage is not the result of any intention on the part of the moss. Sphagnum does not grow in order to store carbon. It grows because it is well adapted to the conditions of waterlogged, nutrient-poor environments, and the carbon storage is a consequence, unintended automatic, of its structural and chemical properties interacting with the waterlogged conditions that prevent decomposition. The bogs that sphagnum creates are distinctive places. They tend to be nutrient-poor, because the acidification that sphagnum creates reduces nutrient availability, and because the waterlogged conditions prevent the flow of nutrient-rich groundwater through the system. The plants that can survive in this environment are specialized ones. Carnivorous plants like sundews and pitcher plants, which supplement their mineral nutrition by trapping and digesting insects, ericaceous shrubs like heather and bog rosemary, which have adaptations to nutrient-poor conditions, sedges and cotton grasses that can tolerate waterlogged soil, and of course, sphagnum itself, growing continuously upward, adding to the peat below, maintaining the conditions that keep the system intact. The water table in an active sphagnum bog is typically very close to the surface, sometimes literally at the surface, so that walking across a bog means walking on a mat of living sphagnum that is essentially floating on water. The living layer of sphagnum at the surface may be only a thin film above largely saturated peat. The moss is growing upward continuously while the material below it slowly accumulates. In the bog's permanent saturation, some remarkable things have been preserved. Objects and organisms that fell into bogs and were buried in the anoxic peat, where the absence of oxygen, combined with the acidic conditions, inhibits decomposition even further, have been preserved for very long periods. Pollen grains recovered from peat deposits allow scientists to reconstruct the history of vegetation and climate in a region going back thousands of years. In some cases, larger organic materials, with seeds, and in certain well-known instances, the remains of humans, have been preserved in remarkable condition in the permanently waterlogged, acidic environment of the peat. A bog in late summer, when the water table drops slightly and the sphagnum surface is drier and more accessible, is a distinctive landscape to walk across. The colors are not the bright greens of well-watered moss, but a palette of muted russets, golds, and olive greens. The colors of sphagnum in various states of hydration and different species mixes. The ground gives slightly with each step, and there is sometimes an audible sound, a soft, yielding compression, as though the ground itself were something alive and responsive, which in a very real sense it is. The air is still, and there is a particular quality of light in a treeless bog, open, without shadow, somewhat melancholy in the way that flat, open landscapes often are. Sphagnum bogs tend to develop in areas where rainfall exceeds evaporation over the course of a year, where the water table stays close to the surface year-round. They are most common in temperate and boreal regions with cool, moist climates, northern Europe, Canada, Siberia, parts of Southeast Asia. But sphagnum itself is a diverse genus with many species, and different species dominate different types of wetland across a wide range of climates. The ecology of peatlands is complex, and research into the interrelationships of the organisms that inhabit them, the plants, the invertebrates, the specialized microbial communities that process organic matter even in these challenging conditions, is ongoing. What is clear is that peatlands represent a distinctive ecosystem type, one whose existence is made possible by the particular biology of sphagnum moss, and whose characteristics are unlike those of almost any other terrestrial ecosystem. All of this, beginning with a plant so small that you can hold a handful of it in your palm, and it feels like holding a small, cool, wet world. Most plants, when they lose too much water, die. The damage done to their cells by dehydration, the collapse of membranes, the concentration of solutes to toxic levels, the disruption of cellular machinery, is irreversible. A wilting plant that does not receive water in time will not recover. This is the vulnerability of being vascular and rooted. A plant is committed to maintaining its hydrated state, and if it cannot do so, it dies. Moss has a different relationship with drying. Many mosses can lose a very large percentage of their cellular water, sometimes the overwhelming majority of it, and yet remain alive. Their cells survive in a severely desiccated state, metabolic activity slowed to nearly zero, waiting for water to return. When it does, when rain falls, when dew forms, when humidity rises, they rehydrate and resume metabolic activity as though nothing had happened. This property is called desiccation tolerance, and it is one of the most remarkable features of moss biology. To understand why this is so extraordinary, it helps to think about what happens to a cell when it dries out. Water is not simply a solvent in which cellular processes happen. It is integral to the structure of proteins, membranes, and other cellular components. Remove the water, and the proteins lose their shape. The membranes collapse, the delicate architecture of the cell is disrupted. Most cells cannot survive this disruption because the damage is too extensive to repair. Desiccation-tolerant mosses have mechanisms that protect their cells during drying and facilitate repair during rehydration. Research in this area is ongoing, and the molecular details vary among species, but several general mechanisms have been identified. Some mosses accumulate molecules that replace the structural role of water during drying, preventing proteins and membranes from collapsing. Others upregulate repair mechanisms as they begin to dry, preparing for the damage they know is coming. Some produce antioxidants that protect against the reactive oxygen species generated during rapid rehydration. The study of desiccation tolerance in mosses is scientifically interesting well beyond the context of moss biology itself. Understanding how cells survive extreme dehydration has potential relevance to questions in medicine, agriculture, and the preservation of biological materials. Research into the genes and proteins involved in desiccation tolerance in mosses has identified some molecular mechanisms that appear to be conserved across a wide range of organisms, suggesting that the fundamental biology of surviving desiccation is ancient and widely shared. But in the everyday world of a forest or a rock face, desiccation tolerance appears more simply as this. The moss that was crisp and dull and almost lifeless yesterday, after the long dry spell, is soft and bright and fully functional today, after last night's rain. Watch it happen in time-lapse, if you have access to such footage. The dried moss colony, viewed from above, resembles something between brown paper and dried herbs. Dull, contracted with the leaves curled tightly against the stems to minimize water loss, the whole surface appearing almost dead. Then the rain arrives. Within minutes, the leaves begin to uncurl. Color deepens toward the rich green of full hydration. By the time an hour has passed, the colony looks entirely different. Plump, bright, responsive, alive in a way it did not appear to be before. The speed of this transformation is surprising to anyone who does not expect it. This capacity for dormancy and revival shapes the ecological behavior of mosses in important ways. Mosses can colonize environments where other plants could not survive. Exposed rock faces that bake dry in summer and are saturated in winter, south-facing slopes that experience regular drying cycles, environments where water availability is irregular or unpredictable. The desiccation-tolerant mosses treat dry periods not as catastrophes but as pauses, temporary interruptions in activity from which recovery is expected and routine. Not all mosses are equally desiccation-tolerant. Species that grow in permanently moist environments, stream margin mosses, mosses in the spray of waterfalls, tend to have lower desiccation tolerance than species adapted to more exposed, intermittently dry conditions. Evolution has calibrated each species' tolerance to the conditions it is most likely to encounter in its typical habitat. There is also a difference between what might be called slow desiccation and rapid desiccation. Many mosses that can survive slow, gradual drying are nonetheless damaged by rapid dehydration. The cell protection mechanisms are inducible responses that require time to deploy. A moss that is dried slowly over hours or days may survive intact. The same moss dried quickly over minutes may suffer more significant damage, even if it ultimately recovers. This has practical implications for how mosses respond to sudden weather events versus gradual seasonal drying. The dormancy itself, the state of minimal metabolic activity during severe desiccation, is poorly understood in its details. What happens inside the cells of a dormant, desiccated moss? What processes are maintained, however minimally, and which are suspended? How does the plant recognize when conditions have changed and it is safe to rehydrate? These are questions that research is beginning to address, but much remains uncertain. What is clear is that dormancy is not death. The desiccated moss that looks, to a casual glance, like dead plant material is not dead. It is waiting. The word waiting implies consciousness and intention, which we should not attribute to the moss. But it is an apt description of the observable state. Suspended animation, readiness to resume, all processes paused but not terminated. And when the rain returns, and eventually it always returns, the moss resumes where it left off, without apparent damage, without the need for recovery time or the accumulation of reserves or any visible sign that it was ever anything other than what it now appears to be. Living, green, quiet, patient. This is perhaps one of the qualities that makes moss feel philosophically resonant to those who spend time thinking about it. Not an active, striving resilience, not the dramatic comeback of a storm-battered tree that slowly regrows its broken branches, but a still, passive capacity to endure, to wait, to continue. A resilience that resembles acceptance more than resistance. We're not the moss, but there is something worth noticing, perhaps in the simplicity of that strategy. When conditions are unfavorable, reduce activity, wait, and be ready to resume when they change. The conventional image of moss, soft green, growing in the damp shade of a forest floor, captures something true about the preferences of many moss species. But it misses the full geographic and environmental range of this plant group, which extends from the tropics to the poles and from permanently submerged stream beds to the sun-baked surfaces of desert soil. Consider Antarctica. It is not a place where, in the popular imagination, anything green grows. And indeed, the ice sheets that cover most of the continent support virtually no plant life. But along the Antarctic Peninsula and on some of the subantarctic islands, where temperatures are slightly less extreme and liquid water is available during the summer months, several species of moss do grow. Antarctic mosses contend with some of the most challenging growing conditions on Earth. Short, cold growing seasons, intense ultraviolet radiation during the austral summer, freeze-thaw cycles that can occur on a daily basis soils, where they exist, that are typically thin, nutrient-poor, and physically unstable. Research on Antarctic mosses has revealed some interesting adaptations to these conditions. Some Antarctic moss species contain high concentrations of pigments that absorb UV radiation and protect the photosynthetic machinery beneath from damage. Some form dense, compressed mats that insulate the interior of the colony from temperature fluctuations, maintaining conditions inside the mat that are somewhat warmer than the external air temperature during the growing season. Studies have measured the temperature inside dense moss mats at significantly higher levels than surrounding air temperatures on sunny days, a consequence of the dark green surface absorbing solar radiation. In Antarctica, moss growth is extraordinarily slow. A moss bank that appears to be a few centimeters thick may represent decades, or even centuries of accumulated growth. Research on some Antarctic moss banks has suggested that the deepest layers can be quite old, though the precise ages depend on species location and conditions. At the other end of the spectrum, desert environments seem like the last place one would expect to find moss. And in true desert cores, the driest, most extreme desert environments, moss is indeed largely absent. But in the semi-arid regions that border many deserts, and in certain microhabitats within otherwise arid landscapes, the north-facing sides of rocks, the interiors of rock crevices, the shaded soil beneath overhanging ledges, mosses do occur, and some are specifically adapted to tolerate extended dry periods. Desert mosses typically represent the extreme end of desiccation tolerance. They may remain dry for months or years dormant and contracted on the rock surface before a rare rain event triggers rehydration and and a brief burst of metabolic activity, growth, and possibly reproduction. These mosses contribute to the biological soil crusts that are important in arid ecosystems. Communities of cyanobacteria, lichens, mosses, and other organisms that form a thin, fragile skin on desert soil surfaces, contributing to soil stability and nitrogen fixation. The biological soil crust communities of arid lands are particularly sensitive to physical disturbance. When these crusts are broken up by footsteps, by tire tracks, by the hooves of grazing animals, the mosses and other crust organisms are killed, and the protective covering they provided to the soil surface is lost. Recovery of a disturbed biological soil crust can take years, or even decades, in arid environments where conditions that allow crust organisms to grow are limited to infrequent wet periods. This fragility is worth understanding without exaggerating its implications. Biological soil crusts are important components of arid ecosystems, but the ecosystem consequences of their disruption depend heavily on the nature, extent, and duration of the disturbance. Urban environments present a different kind of extreme, and one that mosses have navigated with perhaps surprising success. Cities are, in environmental terms, complicated places. Chemically altered water, from rooftop runoff from road surfaces, altered temperature regimes, the urban heat island effect, reduced humidity in some areas, changed light conditions, surfaces made of novel materials like concrete, metal, asphalt, and glass. And yet mosses grow in cities, on walls, on pavements, in gutters, on the north sides of buildings, on old stone structures. Urban mosses are typically hardy, pollution-tolerant species, not the most diverse or delicate of their group, but representatives of lineages that can cope with the particular stresses of urban environments. They colonize the mortar between bricks, the cracks in concrete, the permanent damp spots beneath downspouts, the microtopography of aged stone surfaces. They are often the first plants to establish on newly available surfaces, and they can persist for years in conditions that would exclude most other plants. There is something touching, though we should be careful not to over-sentimentalize, about moss growing on old gravestones, on ancient stone walls, on the ruins of buildings that humans have long since abandoned. These particular colonizations are a reminder that the biological world does not respect the human distinction between built and natural, and that given sufficient time, the softer processes of life tend to reclaim even the hardest surfaces. From Antarctica to the desert, from the Arctic to the tropics, from pristine wilderness to the center of the city, the geographic and environmental range of mosses reflects their fundamental adaptability. Not adaptability in the dramatic sense of rapid evolutionary change in response to novel conditions, but adaptability in the structural sense, a body plan and a set of physiological properties that happen to be useful in an unexpectedly wide range of environments. The moss that grows on a stone wall in a rainy city and the moss that clings to an Antarctic rock face are not the same species, but they are related, sharing a fundamental body plan and set of physiological strategies that have proven over hundreds of millions of years and across the full range of Earth's environments to be remarkably durable. And they are both quietly doing exactly what moss does, absorbing what water is available, photosynthesizing when light allows, persisting through the difficult periods, growing slowly and steadily whenever conditions permit. Unhurried, unremarkable, everywhere. Moss is not random. Look closely at any moss plant, not at the colony as a whole, but at a single shoot, a single stem, the arrangement of leaves along that stem, and you begin to see structure, pattern, a geometry that is not accidental, but functional. The leaves of most mosses are arranged along their stems in specific patterns. and these patterns vary among species in ways that reflect their ecological requirements and evolutionary history. Some species have leaves arranged in 2 rows on opposite sides of the stem, giving the shoot a flat, fern-like appearance. Others have leaves arranged in 3 rows or in spiral patterns around the stem, creating a more 3-dimensional structure. Still others have leaves that radiate from the stem in all directions, forming a cylindrical structure with no obvious preferred orientation. These arrangements are not arbitrary. The geometry of leaf arrangement affects how the plant captures light and water. A moss growing in low-light conditions may benefit from a flat, 2-rowed leaf arrangement that maximizes the amount of leaf surface exposed to diffuse light arriving from above. A moss growing in a position where light arrives from multiple directions may benefit from a more radially symmetrical arrangement. The architectural geometry of the plant and the ecology of the habitat it occupies are closely related, though the precise relationships are often complex and not fully worked out. The shape of individual moss leaves is also variable and functionally interesting. Many moss leaves come to a fine point, sometimes an extremely fine hair-like point at the tip. This hair point serves multiple functions. In some species, it is thought to help with water capillarity, drawing water toward the leaf. In others, it may help protect the photosynthetic tissue below from excessive light, particularly in exposed habitats. In still others, it may help anchor the plant in the substrate. The same structure, present across many moss species, has been co-opted for different functions in different lineages. The capsules of the sporophyte phase also display considerable geometric variation. Some are cylindrical, some spherical, some flask-shaped, some inclined at angles. The shape of the capsule relates to the mechanism of spore dispersal, the way air moves around and through the capsule opening, the way the peristome teeth open and close in response to humidity, the way spores are directed when they leave the capsule. All of these are influenced by capsule geometry. What looks, at first glance, like simple variation in form, turns out on closer examination to be a record of evolutionary solutions to specific physical problems. At the colony level, different growth forms represent different strategies for occupying space and managing resources. Some mosses form dense, compressed mats, low and tight against the substrate, with little airspace between individuals. This form reduces water loss, provides structural stability, and may allow the colony to hold more water relative to its volume. Other mosses form loose, open mats with more airspace, a form that allows greater light penetration to the interior of the colony, potentially supporting a larger community of associated organisms. Some mosses form cushions, hemispherical domes of tightly packed shoots rising above the substrate surface. The cushion form is particularly common in exposed environments. Rocky summits, cliff faces, ancient walls. The geometry of the cushion has thermal properties. The rounded surface sheds wind. The interior of the cushion is insulated from temperature extremes. The orientation of the dome maximizes solar heating during cold periods. A cushion of moss on an exposed rocky summit may maintain interior temperatures considerably higher than the surrounding air on sunny days, effectively extending the growing season for the plants within. There are also mosses that grow in pendant forms, hanging from branches or rock faces in long feathery streams. These pendant mosses, common in humid cloud forests of tropical and subtropical mountains, may play a role in intercepting fog and cloud moisture. Their long, flowing structure maximizes the surface area available for condensation. In cloud forests, where trees are festooned with these hanging moss curtains, the amount of water intercepted from cloud may be significant in the local water balance. The mathematical principles underlying moss growth patterns are not unique to moss. The spiral arrangements seen in many species follow the same principles that govern the arrangement of seeds in a sunflower or scales on a pine cone. They arise from the way plant growth is regulated at the cellular level. The universe of patterns available to life is constrained by physics and mathematics. When similar problems are solved, how to pack the most photosynthetic surface into the least space, how to maximize water capture, similar solutions sometimes appear. Viewing moss through this lens of geometry and pattern shifts the experience of looking at it. Instead of simply seeing something soft and green and pleasingly textured, you begin to see a record of functional decisions accumulated over evolutionary time. Each arrangement of leaves, each capsule shape, each colony form, a solution to a problem posed by the physical world. This is one of the rewards of sustained, careful attention. What appears simple from a distance reveals structure and logic on closer examination. And what appears complex from a distance is sometimes explicable, if slowly, by physics and evolution working together over time. There is a long history between humans and moss, though it is not a history that features prominently in the stories we tell about nature and civilization. Moss did not feed great populations, or build empires, or give rise to major industries. But it was used in quiet ways by human communities across many cultures and time periods, and it continues to have a place in the human world today. Different in form from its historical uses, but continuous with them in spirit. For much of human history, particularly in cooler northern climates where moss is abundant, the most practical use of moss was as an insulating material. The same properties that allow moss to hold water, its high surface area, the spaces between its cells and between its stems, also make it an effective insulator of a different kind of thermal insulator. Dried moss packed between wall logs in a cabin, stuffed into gaps between planks laid under and over materials being kept cool or warm, provided insulation that was readily available, free, and easily replenished. In Scandinavia, parts of northern Russia, Canada, and other boreal regions, moss was historically used as a stuffing material for bedding, for lining boots and gloves, for packing around windows and doors in winter. Archaeological evidence suggests these uses go back thousands of years. The women of many northern cultures used sphagnum moss as an absorbent material. Its water-holding capacity makes it effective as a natural sponge for a range of practical purposes. Sphagnum moss has also had a documented role in wound care. Historical accounts from multiple cultures describe the use of sphagnum as a dressing for wounds, and during certain historical periods of conflict, when conventional medical supplies were limited, sphagnum was used as a wound packing material. The acidic conditions created by sphagnum's chemistry are hostile to many bacteria, and the moss's absorption capacity keeps wound surfaces dry, properties that may have contributed to its effectiveness as a wound dressing in certain historical contexts. The use of peat, the accumulated product of sphagnum and other organic material in bogs, as a fuel is a history unto itself in some regions. In Ireland, Scotland, and parts of Northern Europe, peat was the primary fuel for home heating and cooking for centuries, cut from bogs in blocks and dried before burning. The social and cultural history of peat-cutting communities is deeply interwoven with the landscapes of the bogs themselves, and the practices of peat harvesting, though far less prevalent today than historically, remain part of the cultural identity of some communities. In horticulture, Peat moss has been widely used for many decades as a soil amendment, seed-starting medium, and moisture-retaining component of potting mixes. Its low pH, high water retention, and freedom from the pathogens and weed seeds that can contaminate garden soils made it a popular choice among commercial growers and home gardeners alike. In recent decades, awareness of the ecological significance of peatlands has prompted horticultural industries and gardeners to explore alternatives to peat, and the commercial horticultural use of peat moss is declining in some regions as alternatives become more available. The cultivation of living moss for aesthetic purposes is a practice with deep roots in Japanese garden culture, where moss is considered an essential element of a well-designed garden space. Moss gardens, spaces where the ground is carpeted entirely in carefully tended moss, with minimal or no presence of other plants, exist as a recognized garden form in Japan, and some of the famous moss gardens of Kyoto have been maintained for centuries. The aesthetic appreciation of moss in Japanese garden culture reflects a sensibility that values the qualities of age, quietude, and the particular beauty of small, textured surfaces, qualities that moss embodies in a way that more dramatic plants do not. In the contemporary context of terrariums and indoor plant culture, moss has found a new audience. Closed terrariums, glass containers holding a small, self-sustaining ecosystem, often feature moss as a primary element, and the practice of creating and maintaining moss terrariums has developed into a significant hobby with its own practitioners, techniques, and aesthetic conventions. The appeal seems to be related to the same qualities that make moss attractive in the garden: its small scale, its textural richness, its association with moisture and shade and quiet, and its relative ease of maintenance once established. Moss is also finding increasing use in ecological restoration contexts, when degraded habitats, particularly peatlands, but also some forest and rocky environments, are being restored to more natural conditions. Moss transplantation and reintroduction can be an important component of the restoration effort. Mosses are often slow to colonize restoration sites on their own, particularly in areas where the species pool has been reduced and the nearest source of colonists is distant. Actively introducing appropriate moss species can help accelerate the development of appropriate vegetation communities and soil conditions. Research into the use of mosses as bioindicators, organisms whose presence or absence, or whose physiological condition, can indicate the quality of the environment, has expanded in recent decades, and we will discuss this further in a subsequent section. But it represents another dimension of the human-moss relationship, using moss not just as a practical material or aesthetic element, but as a tool for understanding and monitoring the health of the environments we depend on. The relationship between humans and moss has never been dramatic or consequential in the way that the relationships between humans and staple crops, domesticated animals, or timber trees have been. But it is old, it is widespread, and it is ongoing. In a quiet way, moss has been part of the human world for a very long time, available at ankle height, wherever it grows, offering whatever it has to offer to those who think to make use of it. Because moss lacks the protective barriers that many vascular plants have evolved, the waxy cuticle that reduces water exchange with the environment, the stomata that can open and close to regulate gas exchange, the bark that protects woody stems from environmental exposure. It is in direct and intimate contact with its surroundings. Its cells are separated from the air, the water, the substrate by very little. What is in the environment enters the moss readily. This directness of contact makes moss sensitive. It responds quickly and visibly to changes in environmental conditions, in ways that can make it useful as an indicator of those conditions. The classic use of mosses and lichens as bioindicators is in the assessment of air quality, particularly in relation to atmospheric nitrogen and sulfur compounds. These compounds, produced by a range of industrial, agricultural, and transport sources, can affect the pH of rain and the chemistry of the water that mosses absorb. Because moss absorbs water and the dissolved substances it carries directly through its leaves, pollutants dissolved in rainwater or deposited from the atmosphere as particles enter the moss's cells quickly. Research has documented the responses of different moss species to different levels of atmospheric pollution. Some species are sensitive to elevated nitrogen levels and decline or disappear in areas with high nitrogen deposition. Others are more tolerant of nitrogen, but sensitive to sulfur compounds. Still others appear to be relatively tolerant of a broad range of atmospheric conditions and tend to persist even in quite polluted environments. By surveying which moss species are present or absent in an area, ecologists can make inferences about the history and current state of atmospheric chemistry in that area. This kind of bioindication is not precise in the way that a chemical sensor is precise. A moss community integrates the conditions it has experienced over years, not just the current moment. A moss that has been declining because of elevated nitrogen deposition will show the effect of that deposition over the period of time it has been growing, not just in the last rainstorm. This integration over time is both a limitation and a strength of biological monitoring. It gives a picture of conditions over time rather than at a single instant, which can be valuable for understanding chronic rather than acute environmental conditions. Mosses can also accumulate trace metals and other pollutants in their tissues. When atmospheric deposition brings heavy metals, lead, cadmium, arsenic, and others to the surface of a moss colony, those metals are absorbed and retained. The concentration of metals in moss tissue can be measured analytically and compared to reference values to assess the level of metal deposition at a given location. This property has been used in environmental monitoring programs in various countries, where the collection and analysis of moss samples from standardized locations across a region allows the construction of maps of metal deposition patterns. The use of moss in environmental monitoring is an established practice in Europe and elsewhere, and the scientific literature on the subject is substantial. Moss monitoring programs have contributed to our understanding of how atmospheric deposition patterns have changed over time in response to changes in industrial activity, regulatory interventions, and the use of cleaner technologies. It is worth being clear about what moss monitoring can and cannot tell us. It can tell us about the deposition of certain types of pollutants, those that are soluble in water or deposited as particles that can be captured on moss surfaces. It cannot tell us directly about all forms of air pollution, or about the air quality that humans breathe, rather than the air quality affecting moss-level surfaces. It is one tool among many in the assessment of environmental quality, and it is most informative when used alongside other monitoring approaches. The sensitivity of moss to water quality also makes it relevant in freshwater monitoring contexts. Aquatic mosses, species adapted to grow in and around streams, rivers, and lakes, respond to the chemistry of the water they are immersed in. Their presence or absence and the condition of the moss tissue can reflect the nutrient status and chemical characteristics of the water. Research into the use of aquatic mosses as indicators of water quality has been conducted in various freshwater habitats. There is something philosophically interesting, if not scientifically precise, about the idea of a plant that registers its environment so directly, so intimately, so completely. No buffer, no mediation. The world enters the moss as it is. What is in the air and the rain and the water becomes part of what is in the moss. The moss is, in a sense, a continuous record of its own environmental experience, a living account of the conditions it has lived in, written not in symbols, but in the chemical composition of its cells. We use instruments to measure what is in the environment because our own senses are limited, and we want precision. But we are not entirely unlike the moss in one respect. We too are shaped by our environments in ways we may not always notice, accumulating the conditions of our lives and our bodies over time. The difference is that the moss cannot reflect on this, cannot choose to respond differently, cannot move to a better location. It receives what it receives, and it either persists or it does not. The instruments we use to monitor environmental conditions are, in a sense, artificial mosses, devices that tell us what the moss already knows. There is a question worth sitting with separate from any scientific inquiry. Why do we fail to notice small things? Not always, and not everyone. Children, for instance, often notice small things with a vividness that adults have largely lost, pausing over an ant on the pavement or a mushroom at the base of a tree with genuine attention. And there are scientists and naturalists whose entire professional life is organized around the careful observation of small things. But for most adults, in the ordinary flow of daily life, the small world goes largely unseen. Part of this is surely a matter of scale calibration. We live in bodies of a particular size, moving through environments that our senses evolved to process at a particular scale. The scale of moss, millimeters, not meters, is simply outside the range of normal human attention. We notice what affects us, what moves at our pace, what competes or cooperates with us in ways we can perceive. Moss does none of these things in any way that registers on ordinary human perception. It simply grows quietly at ankle height, at the margins of our awareness. Part of it is also the speed of moss, not the speed of its movement. Moss does not move in any way that is visible, but the speed of its change. A moss colony that you saw last year will look essentially the same this year, and almost the same in 5 years. The changes happening in it, growth, reproduction, the slow accumulation of organic matter, the subtle shifts in species composition as conditions change, are not visible on human timescales. We are a species adapted to notice change, and things that do not change quickly seem to fall below the threshold of our attention. And then there is the larger issue of what our culture teaches us to value. In the long tradition of Western nature writing and natural history, size and drama have tended to claim the attention, the grand landscape, the large mammal, the dramatic weather event. The quiet world of small organisms, complex and intricate and significant as it is, does not fit comfortably into the narrative conventions of drama and spectacle that have organized much of how we tell stories about nature. But the scientific story of the living world, the story that emerges from careful observation and measurement and analysis, is not primarily a story of the large and the dramatic. It is in many ways a story of the small and the slow. The majority of species on Earth are organisms that most people have never seen and could not identify: beetles, nematodes, fungi, bacteria, archaea, mosses, lichens. The majority of the biochemical work that keeps the planet's ecosystems functioning is done by organisms that require a microscope to see. The vast majority of the evolutionary history of life is represented by organisms with no common names in most human languages. This is not a complaint about what we pay attention to. It is simply an observation about what the scientific picture of the living world looks like when you take it whole. And taking it whole means including the moss. There is something else to consider: the labor of ecosystem maintenance. Ecosystems do not maintain themselves through dramatic events. They maintain themselves through continuous, undramatic processes: decomposition, nutrient cycling, water retention, soil building, pollination, seed dispersal, most of which are performed by small organisms in ways that are invisible at the scale of normal human perception. The continuity of the living world depends on work that goes largely unnoticed, because it is never interrupted, never dramatic, never calls attention to itself. Moss performs some of that work, holding soil in place, retaining water, building organic matter, providing habitat for the small organisms that perform other parts of the work, contributing slowly and in small increments to the conditions that make larger life possible. None of this is performed for recognition. The moss does not know it is being helpful. There is no awareness in a moss colony, no intentionality, no goal. There is only the continuation of chemical processes that have been operating continuously for hundreds of millions of years, in changing forms, but with unchanged underlying logic. Perhaps the most honest thing we can say about moss is this: it is more ancient than our ability to think about it, more widespread than our ability to track it, more significant than our tendency to notice it, and more patient than our capacity to observe. And it is doing exactly what it has always done, right now beneath the trees and on the rocks and in the crevices of old walls and across the quiet bogs, absorbing what comes to it, contributing what it can, persisting in the way that only things without ambition can persist, completely, simply, without end. Bryology, the scientific study of bryophytes, the group that includes mosses, liverworts, and hornworts, is a field with a long history, and increasingly a lively present. The questions being asked of mosses and their relatives today extend from the fundamental to the applied, and the tools available to answer them have changed enormously in recent decades. One of the most active areas of current research concerns the genetics of desiccation tolerance. We have discussed the remarkable ability of many mosses to survive severe dehydration and recover upon rehydration. And we have noted that the molecular mechanisms underlying this ability are not fully understood. Research groups around the world are working to identify the specific genes that are activated or suppressed during the desiccation process, and to understand how these genes interact with the cellular machinery to protect and repair cellular structures during drying and recovery. This research has both basic and applied dimensions. On the basic science side, understanding desiccation tolerance in mosses contributes to our understanding of how cells manage stress more generally, a fundamental question in cell biology. On the applied side, there is interest in whether the genes and mechanisms involved in moss desiccation tolerance could have applications in agriculture, where crops in water-limited environments could potentially benefit from enhanced drought tolerance. This is speculative and long-range. The path from identifying genes in mosses to developing drought-tolerant crops is long and uncertain, but the research is proceeding. Another area of active research concerns the evolutionary position of bryophytes in the plant family tree. This might seem like a settled question. Mosses are clearly among the earliest land plants, but the details of which lineage of bryophytes diverged first from the ancestor of all land plants remain a subject of scientific discussion. Different analyses of different datasets have produced somewhat different conclusions about the relationships among mosses, liverworts, and hornworts, and how these groups are related to the vascular plants. Advances in genomic sequencing and phylogenetic analysis are gradually refining our understanding of these relationships, but with some nuance that is likely to continue to be debated in the scientific literature for some time. The ecology of moss communities, how they are assembled, what determines which species occur where, how they change over time, how they interact with other components of the ecosystems they inhabit, is an area where much remains to be learned. Large-scale datasets on moss distribution and abundance, combined with environmental data, are allowing ecologists to examine patterns and processes at scales that were not previously possible. Long-term monitoring studies are beginning to document how moss communities change over decades in response to changing conditions. In the context of peatland ecology, there is significant research effort focused on understanding how peatland ecosystems function, how carbon is cycled, how water moves through peat, how the plant and microbial communities interact, and how these systems respond to environmental changes. This research has implications for the management and conservation of existing peatlands, as well as for efforts to restore degraded peatlands to more natural conditions. Peatland restoration is itself an active practical field. Many peatlands around the world have been drained for agriculture, forestry, or peat extraction, and the ecological consequences of this drainage include changes in plant communities, altered water and carbon cycling, and in some cases ongoing carbon release. Restoring drained peatlands involves re-wetting them, blocking drainage channels, allowing water tables to rise, and in many cases, actively reintroducing sphagnum and other characteristic peatland vegetation. Research into the most effective techniques for peatland restoration and into the recovery of ecological functions after restoration is ongoing. The use of mosses in urban greening and green infrastructure is an area of growing practical interest. Moss walls and moss panels, surfaces covered with living moss maintained by controlled moisture systems, have been used in some urban contexts as aesthetic features, and in some cases, as a means of improving air quality by capturing particulate matter. The science behind the air quality benefits of moss walls is more nuanced than it is sometimes presented, and research is working to understand the conditions under which such installations might provide meaningful benefits, and the conditions under which they might not. Biological taxonomy. The classification and naming of mosses and their relatives continues as a field. New species are still being described, particularly from understudied regions of the tropics, where the full diversity of the bryophyte flora is not yet documented. Molecular tools have also led to the reclassification of some previously described species, as genetic analysis has revealed that what appeared to be single species based on morphology sometimes turns out to be multiple distinct lineages. There is also a citizen science dimension developing in bryology, as interest in moss among non-specialist naturalists has grown. Apps and online platforms that allow the identification and recording of species observations are beginning to generate large data sets of bryophyte occurrence records contributed by non-specialists. And these data sets, while requiring careful quality control, have the potential to supplement the information available from more targeted scientific surveys. All of this research proceeds with the characteristic pace of science, incremental, careful, subject to revision as new evidence accumulates. The picture of mosses that emerges from this ongoing inquiry is not dramatic, and it is not complete, and it will continue to change as new questions are asked and new tools become available to answer them. What it represents is the ongoing effort to know more precisely and more completely what has been living at ankle height, unnoticed, for 450 million years. The rain has been falling for several hours, and now it has stopped. In the forest, the sound changes. The steady percussion of drops on leaves, on bark, on the soft surface of the forest floor, fades, and is replaced by a different kind of sound. The sound of water dripping from leaf to leaf through the canopy, irregular and diminishing, each drop a few seconds behind the last, the forest letting go of the rain it has caught and held. By the time the dripping slows and finally stops, the forest floor is saturated. The soil beneath the moss is dark with moisture, the mineral grains held apart by water films, the fungal networks threaded through the soil drawing in what they need. And the moss itself, the great green carpet of it, covering every rock and log and patch of bare soil, is plump and bright and alive in the way that it can only be when it is fully hydrated, fully itself. Kneel here, not as a gesture, not for the sake of metaphor, but simply as a physical act of attention. Lower yourself to the level of the moss and look at it from the side, at a shallow angle, in the last of the afternoon light that is now returning as the clouds begin to thin and separate. The droplets of water are everywhere. They rest on the tips of leaves, balanced there as though placed with care, each one a small sphere of perfect transparency. They hang from the ends of sporophyte capsule stalks. They collect in the miniature bowls formed by the curled edges of certain leaves, tiny cups no larger than pinheads, each one filled to its brim with clean rainwater. In each of these droplets, if you could look closely enough, you would see the reflected image of whatever is above it, the canopy of leaves, the pale sky, the silhouette of a bird moving through the branches. The moss carries the sky in its water droplets. Every leaf that curves correctly, every stem that bends at the right angle, holds a piece of the world above reflected and contained. And in the soil beneath the moss, and in the water that is now slowly percolating downward through the moss tissue and into the mineral soil below, processes are continuing that have no dramatic names and produce no visible effects. Bacteria are taking up nitrogen. Fungal hyphae are exchanging resources with plant roots. Dead moss tissue is being slowly decomposed, its carbon and nitrogen released back into the chemical cycle that moves materials through the ecosystem continuously, without interruption, without rest. The moss does not observe any of this. It does not know that it is part of a process larger than itself. It absorbs what arrives, it photosynthesizes while light is available. It grows in the direction that growth is possible. It prepares, in its own unhurried way, for the next reproductive opportunity, the next dry period, the next rain. It does not rush. It does not compete for height. When the trees above extend their canopies across the sky and cut off the direct light, the moss does not struggle against this. It adapts to the available light, uses what arrives, and continues. When drought comes and the moss dries and contracts against the stone, it does not resist. It waits. When water returns, it resumes. This is not immoral, exactly. The moss is not a model for human behavior in any direct or prescriptive sense. It is a living organism with no consciousness, no intention, no capacity for the kind of reflection that would allow it to consider what it is doing. It simply is what it is, and does what it does in the way that all living things simply are and do. But there is something worth noticing in the fact that the quietest, most overlooked plants on Earth have been here longer than any other land plant lineage, have survived every mass extinction and every dramatic climate change of the past 400 million years, have colonized every continent and nearly every terrestrial environment on Earth, and are still here, right now, doing what they have always done. Not through drama, not through dominance, not through the conquest of height or the accumulation of mass, through patience, through the efficient use of what is available, through the capacity to endure the unfavorable and resume during the favorable, through the consistent, unremarkable, continuous performance of the basic functions of life. The evening light is changing as the last clouds move away to the east. The angle of the sun through the trees shifts, and the moss is briefly illuminated in a particular way, the green of it taking on a warmth it doesn't have in flat light, the texture of it suddenly visible as a pattern of shadow and highlight. each tiny leaf tip catching the light separately so that the entire colony shimmers slightly like something breathing. And then the light shifts again, and the moss returns to its ordinary appearance, soft green, still, covering the rock and the log and the soil with the unhurried thoroughness of something that has been growing here for longer than anyone can meaningfully imagine. Beneath every forest step, under the leaf litter, under the snow, under the seasons turning, a quiet green world, continuing, softly, without end,